नहीं पूछेगा आपके पास? अरे सर नहीं, मैं भेज रहा हूँ ना, मुझे पता ही नहीं है। जी जी सर। सर आप जब भी जुड़ जाएंगे आपको करा देंगे, है ना, जल्दी से जल्दी मैं भेजता हूँ।
Namaskar, Swagatam. We would like to welcome you all on AYUSH SAMRIDDHI International Web Conference Series 499th journey. The journey which was started during COVID pandemic for knowledge sharing, brainstorming on exclusive points, for celebrating each and every day with relation to AYUSH and our health. So I am really sincerely thankful to you all for your mentorship, support and guidance, due to which we are today eager to touch 500th milestone after this webinar, with webinar of scope and practice of AYUSH in the Netherlands. That is today, and tomorrow on 501st webinar, that will be a grand webinar, I hope, with 51 speakers blessing us on strategy for global awareness of AYUSH. Thank you very much. Thank you, Vishnu Prasad Sharma ji. Thank you, Agnihotri sir. Thank you, Harish Verma ji from Canada. Thank you, Andrea ji. Thank you very much. Meena Ahuja ji, and so on, all experts joining me on the dais, off the dais. Today's speaker selection, the sequence, the expertise, everything is communicated by Dr. Akhilesh Bhargav. He is head of surgery department in his college, Government Astang Ayurved College, and active in Nasha Mukti also, palliative care also. So I would like to welcome Professor Vishnu Prasad Sharma ji for his initial blessings.
Namaskar. Good evening to everybody. Respected Professor V.K. Agnihotri ji, respected Professor Harish Verma ji, Dr. Pavan Kumar Sharma ji, distinguished guests, eminent speaker, ladies and gentlemen. I, Professor Dr. Vishnu Prasad Sharma, welcome all of you to today's evening webinar on AYUSH safe and effective antimicrobials. It is an important subject in this webinar. In this webinar, we have eminent speaker, Dr. Ashok Thakur, Associate Professor, Department of Medicine, MGM Medical College, Indore. I welcome you. Dr. Raghavendra Dubey, Head of Institute of Pharmaceutical Sciences, SAG University, Indore. I welcome you. Next, I want to welcome Dr. Ritu Kapoor from Jodhpur. I welcome you. Next, Dr. Manoj Kumar from Jodhpur. I welcome you, sir. Next, I want to welcome Dr. Akhilesh Bhargav from Jodhpur to this evening webinar on AYUSH safe and effective antimicrobials. So once again, I welcome all the eminent speaker, distinguished guests, ladies and gentlemen, to this evening webinar on AYUSH safe and effective antimicrobial. Thanks. Namaskar. Over to Pavan ji.
Thank you very much, sir. I would like to request you all, while starting this webinar, please see your WhatsApp also, because from 9 p.m. we are going to start the Netherlands scope and practice of AYUSH in Netherlands. YouTube link will be different, but Zoom link will be same. So please stay with us for that a new subject, new country. Thank you. So Agnihotri ji.
Namaskar. Thank you, Dr. Pawan Sharma ji, and welcome to all our guest speakers of today's webinar, along with all the dignitaries who have joined our platform. As we are having our 499th webinar on today evening with the topic of AYUSH safe and effective antimicrobials. So the idea in AYUSH, especially in Ayurveda, as we have got 8 steps of Ayurveda, Ashtanga Ayurveda, we have got one step or one part which is known as the Bhuti Vidya. And for Bhut, we think we understood as things which are not seen by eyes, or in other words, we can say which has passed, which has been already taken place. So, in the reference of Bhut, or in the reference of microbials, which are not seen by the naked eye, it's very important to know that in those days, in the era of say 5,000 years back, how this microbials effect was there on human body or the living beings, and what was the remedy with the Ayurvedic persons. So this microbials, whenever we talk about the microbials, we will talk for the antimicrobials. So for antimicrobials, what we understood that all the rust preparations which are having Paradgandha preparation, or they are known as rasas, they are our said to be the antibiotics or our antimicrobials. Apart from Nim and other things which are having their Katkashai rasas, they are and they had been effective and they are effective in a formulation of Arogvardnivati or Kaishor Gukulu or whatsoever. So today we are going to have two great speakers from Jodhpur University and two great speakers from Indore. And when we are talking about the antimicrobials, very naturally it comes the name of the cancer or cancer cells and effect of any Ayurvedic medicine over the cancer cells. And Dr. Akhilesh Kumar Bhargava is very much with us, by whom, as Professor Pawan Sharma told, that it is all designed and selected by Dr. Akhilesh Bhargav ji. So today evening we are going to listen a lot of things regarding the antimicrobials in special reference to our AYUSH system. Or we would think that this evening our guest speakers are going to enhance our knowledge in all that sector where the Ayurveda can do anything better. Of course, homeopathy has also got some very good means effect, as we were having on that stomach cancer day. Our Tripathi or Ji was telling something. So I think today also we are going to have a very interesting webinar, and with no delay, we would like to have their good and expertise and inputs, because we have got another seminar of 500 say target that is on the Netherlands day. So we are having a tough time or the short of time. So with no say wastage of time, we would like to hand over this platform to Professor Pawan Sharma ji, so that he will continue the next proceedings. Thank you so much. Over to Pawan Sharma ji.
Thank you very much, sir. Without wasting much time, today I would like to welcome Dr. Ashok Thakur. Welcome, Dr. Ashok ji. Welcome. And now I would like to hand over the charge of moderation to Dr. Akhilesh Kumar Bhargav, my dear brother and friend from Ashtanga Ayurveda
Thank
College.
you, sir. Thank you.
Welcome.
Thank you very much, Professor Pawan ji sir, Professor Agnihotri ji, Vishnu Prasad Sharma ji, for being a part of IRES 499 webinar. Welcome to all of you in the antimicrobial seminar, time needed discussion and very important part of today. आज का जो विषय है वो बहुत इम्पोर्टेंट है डॉक्टर्स के लिए भी और पेशेंट्स के लिए भी। आज सभी सरकारें इसलिए चिंतित हैं कि जो भी एंटीबायोटिक्स पेशेंट्स को दी जा रही हैं, क्या वो डिजीज के लिए उपयुक्त हैं, क्या वो सही मात्रा में दी जा रही हैं, क्या वो टाइम नीडेड है, क्या उनको मरीजों की आवश्यकता है, इन सब पर विचार करना आवश्यकता है। और सरकारें भी इस बारे में प्रयास कर रही हैं। डॉक्टर्स की भी इथिक्स है कि वो पेशेंट को किस तरीके से ट्रीट करते हैं, क्योंकि पेशेंट को प्रिस्क्रिप्शन पे जो मेडिसिन लिखी होती है वो उसे लेने के लिए बाध्यता होती है, आज इस अवसर पर हम सभी मिलकर के इस चर्चा को आगे बढ़ाएंगे कि किस प्रकार आयुर्वेदिक एंटीबायोटिक्स आर वेरी यूजफुल इन द ट्रीटमेंट ऑफ ए पेशेंट. So first of all, I am inviting our learned speaker, Dr. अशोक जी ठाकुर साहब, एसोसिएट प्रोफेसर MGM मेडिकल कॉलेज इंदौर. आप इंदौर के मेडिकल कॉलेज में एसोसिएट प्रोफेसर हैं मेडिसिन डिपार्टमेंट में. इसके अलावा आप स्थानीय मेडिकल टीचर्स एसोसिएशन जो है मेडिकल कॉलेज की, उसमें आप सचिव भी हैं. पूर्व में आप इंदौर महानगर के अध्यक्ष भी रहे हैं आरोग्य भारती के. और सबसे बड़ी बात जो हमारे फेवर की है कि आप भारतीय सभ्यता संस्कृति के समर्थक है एवं आयुर्वेद पर आपने अनेक सेमिनारों में अपने लेक्चर भी दिए हैं और इंदौर में आपकी एक प्रतिष्ठा है, आपको सम्मान के साथ देखा जाता है और आज ये मंच आपको यहाँ पर पाकर हर्षित है. अगेन आई एम इनवाइटिंग डॉक्टर अशोक ठाकुर फॉर गिव ए वैल्यूएबल सजेशन फॉर द एंटीबायोटिक्स इन प्रेजेंट era.
Thank you, Dr. Akhilesh ji. Basically, you had chosen a very great topic and a topic which is of great consideration in modern medicine and in AYUSH medicines both. So I want to make a presentation. Should I share my screen or it will be presented by some other else?
No, no, sir, we cannot see. हम नहीं देख पा रहे हैं. आप शेयर कर पा रहे हैं?
You can.
I am trying to share. I am not sharing. I am now trying to share. Microsoft Photo document camera whiteboard.
We have made you co-host.
Okay, so I'm trying to share with camera whiteboard screen. Start recording. Save. One minute, sir. I'm just trying to share. I think it is not getting shared. Google Drive, documents, camera, whiteboard, screen. Start now.
Sir, if it is any difficulty, you can WhatsApp your PPT stuff to either.
I had WhatsApped it to Akhilesh ji. Can
yes, yeah.
Akhilesh ji, आप try करिए ना share करने.
Yes, sir, definitely. Pawan sir, I have sent the email of presentation, Dr. Thakur sir.
And you are also co-host. You can do by your own.
I'm trying, sir. I am not able to do it.
Okay.
So there is some problem with me to share the PPT. Professor Pawan sir, please try to share the PPT. Pawan sir. Pawan sir. तो नहीं क्या सर? पवन सर.
तो नहीं, he might be in another.
सर सुन नहीं पा रहे हैं मुझे.
हाँ.
सर को मैंने पीपीटी कर दिया था.
अच्छा अच्छा. हाँ, yes. He is mute. He is there.
ठाकुर सर,
ये दिख रहा है क्या?
नो सर.
आप देख लीजिए, इनकी पीपीटी आपको मेल किया है इन्होंने, भार्गव जी ने, अगर आप चला सके.
आपने किस ईमेल आईडी पे किया था, यही खोज रहा था.
ठाकुर सर, आपने मेल किया था क्या?
हाँ, आपने जो ईमेल आईडी दी थी ना, उस पे भेजा है मैंने.
हाँ सर, आपको भेजा है.
किस ईमेल आईडी पे कृपया बता दें मुझे.
आयुष समृद्धि मेड फॉर्मर. यस,
हाँ, आयुष समृद्धि पे भेजा है सर.
ठाकुर सर, अपन वर्बली स्टार्ट करते हैं.
हाँ, मैं शुरू करता हूँ जब तक, और सर आप जब तक कोशिश करिए. सो बेसिकली फर्स्ट वी हैव टू अंडरस्टैंड दैट व्हाट इज एंटीमाइक्रोबियल्स. सो एंटीमाइक्रोबियल्स इंक्लूड्स एंटीबायोटिक्स, एंटीवायरल्स, antifungals and antiparasitic medicines used to prevent and treat infections in human, animals and plants. So basically, in a nutshell, we can say that it is effective against all the microorganisms which will produce disease in humans, animals, and plants. So a brief history. I think the presentation is visible now.
Yes, yes, sir. Thank you, Professor Paul, sir.
Sir, next slide, please, sir. So this is the definition which I was stating, sir. Next slide. Now I came to history of antimicrobials. So basically, we all know that antimicrobials revolutionize this treatment, modern treatment and modern medicine. By use of antimicrobials, there is a new era in the treatment modality, and the term antibiotic originally was used to denote the formulations derived from living organisms, but is now used for partially or wholly synthetic antimicrobials. So as you all know that first antibiotic which was discovered by Sir Alexander Fleming in 1928, and it was basically discovered from a mold, a killed mold, and from this he derived this penicillium. And penicillin was the first antibiotic discovered, and it changed all the treatment modalities thereafter. Next slide. Next slide, sir. Thank you, sir. So in this slide, we can see that how antibiotics evolved with time. So in 1929, penicillin, then penicillin chrysogenum, then in 1943, aminoglycosides, streptomyces griseus. In 1945, tetracycline, chlortetracycline, streptomyces aurifaciens. Then came the era of 1950, in which two new aminoglycosides, vancomycin, amicoplastis orientalis, and glycopeptides were discovered. Similarly, quinolones were also discovered in 1945. Pio 1B, 1C, 2, 3, and 4. Pseudomonas aeruginosa, it was effective against Pseudomonas aeruginosa. Then in 1950, macrolides like erythromycin was discovered, which was effective against Saccharopolypsora erythera. Then in 1958, we had discovered cephalosporins, cephalosporin P and C, and it was derived from Cephalosporium acremonium. Then in 1970 to 1980, carbapenems like this meropenem, ertapenem group was discovered, and it was derived from the Streptomyces cattleya. Then there is a new antibiotic, thienamycin, was discovered in the same year, 1976. Similarly, in 1963, lincomycin was discovered, and it was derived from Streptomyces lincollinis. Fluoroquinolones, ciprofloxacin are the synthetic antibiotics, which were basically invented in 1981. Fluoroquinolone, levofloxacin is also a synthetic antibiotic discovered in 1996. Then oxazolidiones like linezolid are synthetic. These are discovered in year 2000. In year 2005, we have got new fluoroquinolones, that is gemifloxacin, which is synthetic. Retinoids, these are discovered in year 2018. The name are CD437 and CD1530. So these all are basically synthetic antibiotics. So you can see by this slide that antibiotics in the initial era were discovered from the different living organisms, and later on it was new synthetic era of antibiotics came even after 1980. And I want to state here that how penicillin was discovered. So basically, when Sir Alexander Fleming was doing some research in his laboratory, and he has a petri dish in which two different colonies were growing, and he found that with the growth of a mold which was incidental growing in that petri dish, he found that those antimicrobial or those bacterial colonies suppressed. So he thought that there are some chemicals which were secreted by this mold, and which is actually suppressing this growth of bacterial colonies. So by this he get an idea that this is giving some this is producing some chemical, and later he purified and derived penicillin. So it's a very interesting story by which this era evolved, and everybody of us knows now know that there are many diseases that, like we will take example of enteric fever or moti jhara. So basically in moti jhara, we used to taught our student about its natural life cycle, in which we say that there is infection in the GI tract, followed by shedding of the epithelium. Then there is this pea soup diarrhea is there. Later on, we will try to basically act our we try that our immunity will kill all those bacteria with time, and it will take a month time to be cured. And in this we will try to give liquid diets to the patient. That was the original treatment prior to use of antibiotics in case of enteric fever. But with the advent of antibiotics, what we found that all this natural history is just a history, and nowadays we start antibiotics after a proper diagnosis using a new test like serum typhi, IgM and IgG, and previously Vidal test. So by this we diagnose the disease according to the clinical features and laboratory test, and after that we start antimicrobials. Antimicrobials, there is some antimicrobials. Then there is some problem in sharing. Raghavendra Dubey admit. Akhilesh ji slide दिख रही है ना? हाँ, it is visible to me.
Yeah, different. You need not to worry for the admissions of newcomers. Don't worry.
Okay,
that I am
okay,
doing.
okay, sir. Okay.
So
Then after this, it revolutionized the treatment, and patients are getting cure in the very first step of the infection. So that's how the beauty of antibiotics came. And in this slide, which is visible to us, go back to previous slide, please. Yeah. In this slide, we can see that there are different type of antimicrobials, which includes antibiotics, antivirals, antifungals, and antiparasitic. Next slide. So this is a diagram depicting the site of actions of different antimicrobials. Like some antimicrobials acts on the cell wall synthesis, like cycloserine, vancomycin, bacitracin, penicillin, cephalosporin, monobactums, carbapenems. Similarly, in the cell cycle, folic acid metabolism, some antibiotics act on the folic acid metabolism, which prevents the conversion of dihydrofolate to tetrahydrofolate, like trimethoprim and sulfonamides. Similarly, cytoplasmic membrane structure is also attacked by the antibiotics polymyxin and the daptomycin. Some antibiotic acts on the genetic material by having their action on the DNA gyrase, like quinolones, nalidixic acid, ciprofloxacin, and novobiocin. Similarly, RNA elongation is also seen. Then there is a group of antibiotics which prevent DNA-directed RNA polymerase activity, and these are the antibiotics rifampin and streptoviricins, like streptomycin. Then protein synthesis is targeted by its 50S inhibitor, by the antibiotics erythromycin, chloramphenicol, clindamycin, and lincomycin. Then protein synthesis by 30S inhibitors is targeted by tetracyclines, spectinomycin, streptomycin, gentamicin, kanamycin, amikacin, nitrofurantoin. And protein synthesis by transportation RNA is targeted by mupirocin and puromycin. So by this slide, we are able to understand that there are good number of antimicrobials which are targeting different targets, and we know the different targets where they are acting and how they are acting. So this knowledge is useful when we are using two antibiotics simultaneously for synergistic action. Next slide. So two antibiotics which are acting at different targets will definitely give more better results, especially in case we see that anti-tubercular therapy or ART, we do not use single antibiotics, but we use combination antibiotics. Similarly, this is antiviral classification, which includes non-retroviral and anti-retroviral. Non-retroviral antibiotics include this acyclic ACV like guanine, then nucleoside analog inhibit viral synthesis, DNA synthesis, guanine. Then GCV acrylic are guanine nucleoside analog inhibitory activity against all herpes virus, especially active against CMV. Then these are the anti-HSV, anti-herpes simplex virus antiviral. Then we have anti-influenza antiviral like AM, that is this uniquely configured tricyclic amine low concentration inhibit the replication of the LAV. Then OTV, this is a transition state analog of this salicylic acid, a protein selective inhibitor of influenza A and B. Then anti-hepatitis, these are interferons which are potent cytokine that possess antiviral immunomodulatory and anti-proliferative activities. These are synthesized by host cells. Similarly, we are having antiretroviral drugs, as I am talking about the ART, which is used for treatment of HIV. So here we are having drugs like NRTI, that is nucleoside reverse transcriptase inhibitor. Then NNRTI is non-nucleoside reverse transcriptase inhibitor. NRTI includes this zidovudine, that is mnemonic is AZT. It's a thymidine analog which is active against both HIV-1 and HIV-2 virus. Similarly, we are having this lamivudine, which is active against HIV-1, HIV-2 and hepatitis B virus. Then example of NNRTI is efavirenz, which is protease inhibitor like sequinavir, attenavir, this nefrinavir and ritonavir. Then we are having this entry inhibitor like enfuotide, miraviroc and reltegravir. Then multi-class combination products are used, as I stated, which includes efavirenz, lopinavir and this ritonavir. Then we have that zidovudine, efavirenz, lamivudine. So all these combinations are used taking the different mode of actions. Next slide. Next slide, please. Sir, next slide, please.
Next is on the screen, antifungals.
Okay, thank you, sir. Thank you. These antifungals, then you all know that these are the drugs which are active against fungus. And 1 minute. These include antibiotics which are active against fungal, and we all heard the name of this pollen, that is amphotericin B, which was effective against the mucormycosis. And there was a lot. Yes, there was a lot fight for this mucormycosis. This amphotericin B in the mucormycosis endemic. Then we are having nystatin and hemicin. Similarly, heterocyclic benzofuron like glycofulvin, echinocandins, we all know capsofungin, micafungin, and enidufulgin. This is a new antifungal. Then azoles like imidazole, topical preparation like clotrimazole, iconazole, miconazole, oxyconazole, systemic are ketoconazole. And triazoles are fluconazole, itraconazole, voriconazole, posaconazole. Then L-amine, terbinafine. Topical agents like this tolnafate, then undicycline nic acid, benzoic acid, cyclopyroxolamine, butanafine, quinidochlor, and sodium thiosulfate. So all these are antifungals having different type of systemic and topical effects. Then this antiparasitic like antimalarials, anti-helminthic and anti-protozoals. So antiparasitic drugs like imidazole class, which includes thiabendazole, then albendazole, fenbendazole, oxyfenbendazole, oxybendazole. So we all heard these names when we are treating the worm infestations. Then we have this imidazethiozoles. This include a newer antiparasitic drug like levamisole. Then we have another group of tetrahydropyrimidines, which include pyrentel and morentel tartrate. These are less in use basically. Then we have macrocyclic lactones like ivermectin, which we heard in treatment of COVID also. But it is mainly a anti-parasitic drug, not a anti-COVID drug. This is used in the initial phase. Later on, recommendation was taken back. Then we have this deromectin, epiromectin, and moxidectin. Piperazines like piperazine, and another group isoquinolones like praziquantel. Next slide. Then basically Akhilesh ji is talking that what is the disadvantage. So I think the biggest disadvantage is that nowadays when we are seeing the history of antimicrobials invention, so there are lot number of antimicrobials invented before 1980, and a few antibiotics were discovered after 1980. And with the inadvertent use or with overuse of the antimicrobial, a new problem of antimicrobial resistance is coming, and which is really a big problem. So antimicrobial resistance occurs when bacteria, virus, fungi, parasites change over time and no longer respond to the medicines, making infections harder to treat and increasing the risk of disease spread, severe illness, and death. As a result of drug resistance, antibiotics and other antimicrobial medicines became ineffective and infections become increasingly difficult or impossible to treat. Next slide. So antibiotic resistance, we need to know 5 things about this. Antibiotic resistance occurs when germs defeat the drug designed to kill them first. Second point is antibiotic resistance can affect people at any stage of the life. Healthy habits can protect you from infections and help stop germs from spreading. Then this antibiotics save human and animal lives. Anytime antibiotics are used, they can lead to side effects and resistance too. Antibiotic resistance has been found in all regions of the world. Next slide. So this is a flow chart which is stating both the development of the antimicrobials. So in this slide, we can see the right part that all the antimicrobials were discovered. Then in case of resistance, you can see that in early 1940s, there is a resistance in the Staphylococcus aureus by producing penicillinase enzyme, which makes the penicillin less effective. Later on, this MRSA strains evolved in 1961, which was not even affected by the higher antibiotics like our this cephalosporin group. Similarly, in late 2000, we can see that good number of organisms are developing resistance with newer and the newest antibiotic. Especially, I want to put emphasis on this ESBL, that is extended spectrum beta lactamase inhibitor. These are gram-negative bacilli which are very resistant and very difficult to treat, and only few drugs like colistin or polymyxin B are effective against these type of drugs. So it's a big challenge. Next slide. What is antimicrobial resistance a global concern? Why is it's a global concern basically? So the emergence and spread of the drug resistance pathogens have acquired new resistance mechanism leading to antimicrobial resistance continues to threaten our ability to treat common infections. Especially alarming is the rapid global spread of the multi and pain resistance bacteria. In common language, we call them superbugs that cause infections that are not treatable with existing antimicrobial medicines such as antibiotics. Next slide. So the clinical pipeline of new antimicrobials is dry, which I was stating. In 2019, WHO identified 32 antibiotics in clinical development that addressed WHO list of priority pathogens, of which only 6 were classified as innovative. Furthermore, a lack of access to quality antimicrobials remains a major issue. Antibiotics shortage are affecting countries of all levels of development, especially in healthcare systems. Antibiotics are becoming increasing ineffective as drug resistance spreads globally, leading to more difficult to treat infections and death. New antibacterials are urgently needed, for example, to treat carbapenem resistant gram negative bacteria. Bacterial infections is identified in WHO priority pathogen list. However, if people do not change the way antibiotics are used now, these new antibiotics will suffer the same fate as the current ones and become ineffective. Next slide. So the cost of antimicrobial resistance to national economies and their health system is significant, as it affects the productivity of patients and their caretakers through prolonged hospital stays and need of for more expensive and intensive care. Without effective tools for prevention and adequate treatment of drug-resistant infections and improved access to existing and new quality assured antimicrobials, the number of people for whom treatment is failing or who die of infections will increase. Medical procedures such as surgery, including cesarean section, hip replacement, cancer, chemotherapy, and organ transplantation will become riskier. Next slide. So by 2050, antimicrobial resistance could cause a global burden of almost $100 trillion over the world. And we can see in the map that it is affecting mostly the Africa and Asian subcontinent. And in Europe, there are 3,90,000 deaths because per year because of the infections. Next slide. Then how to prevent antibiotic resistance? So there should be antibiotic stewardship. It means antibiotics should be used rationally and in the full dose. Development of new novel treatments, preventive measures should be used. As in our Arogya Bharati, we used to say that first approach should to prevent or prevention. And if after prevention infection occurs, then there will be less burden on the antibiotics. As we know from the previous slide that when we use more antibiotics, there are chances of more antibiotic resistance. So prevention should be used. There can be measures like vaccine and rapid diagnostic techniques which can prevent the emergence of the resistance. Next slide. So what accelerates the emergence and spread of antimicrobial resistance? Antimicrobial resistance naturally over time, usually through the genetic changes. Antimicrobial resistance organisms are found in people, animals, food, plants, and the environment. They can spread from person to person or between people and animals, including from food to animal origin. Next slide. The main drivers of antimicrobial resistance include the misuse and overuse of the antimicrobials. This is very important slide. We have to understand that misuse and overuse of the antimicrobials is a big challenge. Then lack of access to clean water, sanitation, and hygiene for both humans and animals, which will predispose to increased chances of infection and increased use of the antimicrobials. Similarly, poor infection and disease prevention control in healthcare facilities and farms. Then poor access to quality, affordable medicines, vaccines, and diagnostics. Lack of awareness and knowledge and lack of enforcement of legislations. Next slide. So antibiotic resistance, you can see there is antibiotic
थोड़ा
resistance.
टाइम का कंसीडर कीजिएगा डॉक्टर.
Only few slides are left. So we can see in this slide in a nutshell that antimicrobial resistance is not only in humans, but it is in animals, then food industry, agriculture, all the sectors. Next slide. So how bacteria and fungi? Next slide, sir. So these are the mechanisms of development of antimicrobial resistance. I think it's not very important. Next slide, sir. Next slide. Then global action plan on antimicrobial resistance. So globally, countries committed to framework set out in the global action plan 2015 on antimicrobial resistance during the 2015 World Health Assembly, and committed to development and implementation of multi-sectoral national action plan. It was subsequently endorsed by the governing bodies of Food and Agriculture Organization of USA, United Nations, and WHO. Next slide. So to achieve the goal and global action plan set out 5 strategic actions. So to improve the awareness and understanding of the antimicrobial resistance first, then to strengthen the knowledge through surveillance and research, to reduce the incidence of infection, to optimize the use of antimicrobials, and to develop the economic case for sustainable investment that takes account of the needs of all countries, and increase investment in new medicine, diagnostic tools, vaccines, and other interventions. Next slide. So this is about this antimicrobial plan. Basically, all the countries in the world are worried about this, and that's why World Antimicrobial Awareness Week was celebrated. And we do have a committee of pharmacovigilance in our institute also, which will take care of this. And this week is celebrated between 18 to 24th November. Next slide. So exploitation of antimicrobials should be avoided, and discovery of the new antimicrobial should be promoted. Then drug combinations which will prevent the emergence of resistance should be used. Good diagnostic and accurate treatment should be more used than compared to empirical antibiotics. Next slide. I think protect yourself, and we have to, as I stated, that prevention is the best thing. So we have to try to prevent the occurrence of infection. Next slide. Actions for healthcare provider. So healthcare provider should prevent the infection and spread of the germs, then improve the antibiotic prescribing, like using rational antibiotics, and full dose of antibiotic use is very important. Then be alert and take action when there is any resistance. Then try to report and give a proper antibiotic according to culture sensitivity reports. Next slide. Next slide. So that's all from my side, and the main references in my presentation were from WHO, CDC, and from our Indian setup. Thank you, everybody. Thank you.
Thank you, Thakur sir, for giving a valuable information about the history of antibiotics and the medicines about the antifungal, antibacterial, antiviral disease. थैंक यू वेरी मच आपका कीमती समय देने के लिए, और भविष्य में भी आपका समय और साथ इसी प्रकार मिलता रहेगा. बहुत बहुत धन्यवाद आपको. नाउ आई एम इनवाइटिंग डॉक्टर रितु कपूर फ्रॉम राजस्थान आयुर्वेद यूनिवर्सिटी, एचओडी टॉक्सिकोलॉजी डिपार्टमेंट. And actually, at 9 o'clock we are starting other webinar from Netherlands. तो मेरा रिक्वेस्ट है सभी से कि हम 15 से 20 मिनट में अपने प्रेजेंटेशन को लेंगे और शॉर्ट में हम अपने प्रेजेंटेशन को समाप्त करेंगे. तो आई एम इनवाइटिंग डॉक्टर रितु कपूर फ्रॉम राजस्थान.
Good evening to all of you.
Good evening, madam.
Thank you so much. First of all, I want to thank the organizing committee for giving me the chance to express my views on this topic of the antimicrobial plants. So thank you, the members of the organizing committee. I'm very thankful to you. Now I start my presentation. Now, my topic is the
कैमरा नहीं
antimicrobial
आपका ठीक आ
property
रहा,
of
आपका कैमरा नहीं ठीक से आ रहा है, ग्रीन शेडो आ रही है। आप डॉक्टर मनोज अतलखा जी के साथ हैं
जी सर,
थोड़ा सा ऐसा फेस करिए कि आपका इंडिविजुअल आप पर फोकस आया है लाइट का। आप कहीं रिफ्लेक्शन हो रहा है, लगता कहीं मिरर वगैरह है कुछ सामने, क्या ग्रीन बैकग्राउंड आ रहा है।
सर मैं कुछ कहती हूँ सर, स्क्रीन शेयर हो रहा है सर?
No, we cannot see the screen. Okay, we can see the screen.
Now screen is started.
Okay, thank you so much, sir. Is it the reflection is over now? Yeah, it is still there.
Okay, madam. Let's start.
Okay, I'll start with my presentation. Next slide, please. Yes, sir. It's not. So, Dr. Ashok ji has already told many things about the antimicrobial agents and the properties, the brief history of that. And I've told by Dr. Clay that I should not go into the detail of all those because of the shortage of time. So I'll discuss the history and the antimicrobial agents. I'll just brief about the antimicrobial compounds, which are, we all know that these microbial compounds are the compounds which actually prevent and cure the microbial diseases. The microbial diseases may be the bacterial diseases or the So the antimicrobials are those compounds which basically act on these particular the bacteria, the fungal, the viral, and the parasitic diseases, and these compounds cure them and kill or inhibit the growth of bacteria or the viruses and all the microbes. So in my presentation, as I belong to the Ayurvedic field, so I will try to enlighten the antimicrobial property of the plants. So plants, we all know that they contain the phytochemicals, and those phytochemicals are being used since long time, and they are used to cure many of the diseases of the human beings. But with the at the time advances and the new techniques come in, so the new synthetic microbes are being prepared and which are being used very often and without any cautious. Everyone was using the antimicrobes to treat many types of the bacteria without knowing the dose and without following the proper schedule of that. And with that, what happens? Many resistant type of bacteria has been developed, and those resistant bacteria are not responding to the synthetic antimicrobial agents. And also with the synthetic antimicrobial compounds, what is happening? Along with the benefits, I won't say that they are not benefiting us. They are benefiting us, but along with that, many types of other types of side effects are also being presented there. These effects are one of the major side effects, should I say, is reactive oxygen species are there, which is causing many type of health hazards to the human beings. So what is happening with the synthetic antibiotics, that these are causing, these are producing many type of side effects also, and which is not with the phytochemicals or the herbal antimicrobial agents. So it is beneficial to use the natural antimicrobial agents, which can benefit us and will not produce any type of harm also. So medicines are primary medicines to treat infections. We all know that. And along in the with the many type of toxic plants have also been mentioned. So what the antimicrobial properties of those toxic plants have been mentioned by our acharyas. They have said, our acharyas have mentioned in our text that this particular toxic plant is used to treat krimi or jantu or many types of kush. We all know that krimis are related to the many types of worms, or maybe the bacteria or the viruses. And if they are talking of the jantu, this medicine is jantugna. Many types of infectious diseases are being treated by that particular compound. So these herbal medicines are the use of these herbal medicines, and also the toxic herbal medicines are used by our acharyas to treat many type of microbes. So this is in the present time, due to the side effects and due to this resistant bacteria, everyone, even the whole world, is looking forward to towards the Ayurveda to see, to find out the new solution of this resistant bacteria and the new solution of the side effect resistant antimicrobial compounds. And for that, the solution is only the herbal plants or the toxic herbal plants. So, and I talk about the datura. We all know that datura is the toxic, and its seeds cause the toxicity there. If the datura seeds are ingested, many types of toxicity is being presented there. And 90s of the datura poisoning is very famous to everyone. I think everyone knows about the 90s of the datura poisoning. Datura, you all know, maybe you know it by the name of Kanak or Unmad. And the phytochemicals of the datura are daturadiol, hyoscyamin, scopolamine, hyoscyamin, beta-cytosterol, tropine, etc. And these chemicals, what these chemicals do? These are the phytochemicals which can kill or which can lower the growth of the microbes there in the body or in the wound. So this datura is being is used as a antimicrobial agent. And nowadays we all know that to validate our concept, new and new researches are being there, being done there to validate the concept, to accept the concept. So for that, many researches has also been done on this. And in those researches, we have found that the growth inhibition was there with the Erwinia and with Pseudomonas. Antibacterial activity of the extract of the aerial parts of the datura was found against gram-positive bacteria. And also no antibacterial activity was found with Escherichia coli and the Pseudomonas aeruginosa. So the study which Acharyas has already been told that datura is datura katuushrasya prantikare kusthani hanti means it is very effective in kush. And we all know that the causative bacteria present in the all these leptis, Mycobacterium leprae. So our acharyas knew this thing, but and they have already told it many years before that the datura is very effective in the kush, and it can inhibit, and it has the ability to effect there in the nobody has done such as they have told that it's antibacterial activity there on the gram-positive. And now come to the Riosa. Gloriosa, we all know that it is known by the name Langli, and also the Agnishika, Kalihari, and the Aprapatni are its name. Gloriosa or Langli is again the toxic, and when ingested, it produces even the hemorrhagic hemorrhagic gastritis, neurological manifestations, and many symptoms are produced when it is ingested. The pharmaceutical compound in the Langli are processing beta-cytosterol, colchicamide, and these chemical compounds have antibacterial and antifungal activities, which are told by AYUSH many of the years back. And these are now validated when the antifungal sensitivity was expressed by this, and the antifungal activity against Candida albicans, Candida glabrata, Trachytomyces has been proved there, and the mild to moderate antibacterial activities have also been shown there, and chloroform fraction Staphylococcus aureus. So this various back proof now, we all can say that this Langli plant bears the antibacterial and the antifungal activity. So it is very say instead of using your synthetic antimicrobial agent, it's good to move to plant products or the phytochemicals which can be bacterial. The famous formulation of this Langiadivati or Langiadila cyanide, the Casisaditel, which are being used or being applied for the antibacterial and in the heal. Next plant is Calotropis. We all know Calotropis. We have seen that widely grown, and that is known by the name or Alarka, Madar, and the Akar. Its vernacular name, we all know it by some says it has a Madar or Arka. So it also causes we all know that the latex of this plant causes the toxicity when it is dropped into the diet, causes the conjunctivitis and there. And our acharyas have recognized Am I audible?
Audible, madam. I think there is some technical problem. We cannot see the slide. There is some disturbance in your voice.
But this is my voice. Is it okay now,
yes,
sir?
Audible, madam.
The slides are shown there?
Yes. No. I cannot see. If there is problem, we can continue orally presentation.
Yes, sir. I'm doing that. Just a bit, sir. I'm trying to.
Okay.
Is it okay now, sir?
No.
Okay, sir. I'll continue verbally only.
Yes.
So, as I was saying that our acharyas has told the properties of the arc as the creamy kusha. Means it was
slide is visible.
Okay, sir. It is the Acharya has told that it can be used in the treatment of the leprosy, and also it has the homicidal effects. So this research is now validated. Many researches has been done to show the properties of the antibacterial and the anti-fungal properties of the calotropis has been done, which shows that that antibacterial activity of calotropis gigantea against Streptococcus and Lactobacillus acidophilus. And also the extract have revealed its antibacterial potential. Numerous microorganisms such as Bacillus and Bacillus subtilis, Shigella, Pseudomonas, E. coli, etc. Now move to the next. Gunja, we all have Gunja, and it is widely grown everywhere. It is in many also. It is known by the name Rati or Sheetapati. You must have seen the goldsmith measuring the gold of weighing gold the Rati. That is why it's known by the name Rati. So when ingested, it causes the hemolytic problems in the body, many hemorrhages there in the body, and due to which the conversion is there, and the sinus or Gunja toxicity resembles that of viper bite. The fibers present in it are in Sagonal, Ebredin, Ebrol, Pricol, and all these phytochemicals, they also work against the Kandu, which is called by many microorganisms, and it also has the inhibitory effect on the growth of the worms or the crimi. So this study this has this effect of the Gunja, which is told by AYUSH, has also validated by any study, and in those studies prove that the Streptococcus aureus is most sensitive to the leaf extract of Gunja. Also, the extract of the stem and the seed oil present was potent against some of the gram-positive bacteria and the Candida albicans. And similarly, the seed oil has potent antimicrobial activity. So this study has also been validated, and due to this, its antibacterial and properties, it is major content of Javadi and Buddha Bhadras. So basically, I want to tell you that these drugs are the toxic drugs. I am talking about the toxic plants and the toxic drugs. So they have the chemicals cause the toxicity. But if those phytochemicals are a very small dose, so they can cause the toxicity to the microbes and produce the suicidal or the inhibitory actions there, and does and causes a very low toxicity to the cells, and we can say very mild, if even means we can say the mildest effect is there on the cell. So the cells, but growth of the microbes and bacteria is prevented. Next, I talk of next by the name marking that as Dobbies use it and in marking the clothes. So it is also a very important drug, and it is known by the name Belava, very common name Belava, we know by this name. And this Belava, though it is a toxic, and but it causes though it is also toxic, but Acharya Sushruta has told it's used in it has study effect there on the left track, and this study has also been proved by the researchers that it has been one of the researchers has told that the antimicrobial efficacy of the nut oil of Semicarpus anacardium. Next study, essential oil extracted from the nut oil is very effective against these subtilis, Streptococcus aureus, vulgaris, E. coli, Aspergillus niger, Aspergillus fumigates, Candida albicans, and glaberata. Also, a remarkable entry in a pronounced inhibitory zone is seen there with Aspergillus niger. So, this property of can be used to treat many diseases. Next, I talk of Kupilu. Kupilu is, you all know, that it is Nutska, and the common name of it is Kuch, belongs to Loganiaceae family. The chemical compound, the Kuchla, we all know that the Kupilu, the strychnine is very toxic when the seeds of it when cultured ingested, it causes the many types of problems. It affects the CNS and even cause like fluorostonas and astronas. So the conversion imposes the death of the tonic and clonic conversion imposes the death of the person. So we know that it is a very log and bomacin are the main chemical components of this particular plant. About the Kuchla, our Acharya has told that it is Kaf with he Jantu Pramahiti. Means it has its prominent effect on the Jantu. It is killing the many types of the microbes present there at the bones or there. And this nowadays the research which has been done on the Kuchla or the strychnine is antimicrobial activity against the Staphylococcus, Salmonella, Klebsiella pneumonia, Aspergillus niger, AYUSH, flavors, and Salmonella, Aromanus, Hydrophila, and Pseudomonas aeruginosa. And also, it has the most susceptibility to Staphylococcus aureus, which is a gram-positive bacteria, and E. coli and the Klebsiella pneumonia, which are the gram-negative bacteria. Now, I talk the next drug, next toxic plant, which is ARN. Again, for the ARN, we all know that it is a that it very good effect to alleviate the from the body. So iron also has about the iron, it has been stated by our acharyas that it has krimi vinashanam effect. It has a inhibitory effect on the growth of the krimi. It has the subtle effect on the bacteria or on the viruses and the fungi. So it can be used to use as an antimicrobial agent. And it's the researches which has been done on the Daracinus communis to prove that its antimicrobial potentials, the bacteria and the fungal strains, which pneumonia is there, but the maximum inhibition zone against and is the maximum inhibitor. Also shows the activity against all 4 gram-positive negative bacterial strains, that is because of aureus and
Dr. Ritu, we can see your slide, but your voice is not clear.
Voice is not clear.
Yes.
So it's resonating.
Please conclude the topic. If there is some problem in your technical mistake in your internet connection.
Let me check, sir. Just wait for a sec. Let me check.
Yes, we are finishing the topic. I think it is 19 slide.
Sir, is it audible now?
Yes, yes.
Is it clear now?
Yes, yes.
So my presentation is almost about to finish now. Can I start now, sir?
Yes. Continue, madam.
So, the last, not the least toxic drug I'm talking about is Chitrak, and the Chitrak is known by the name Ushnavyal and Cheetah. And as it is also a toxic drug, it also produces many type of toxicity there in the body when taken in the fatal dose or when taken in the excessive amount. But when taken in, when used in the limited quantity or the low quantity, it can have the beneficial effects in the leprosy, and also it can remove the worms from the wounds or from the body. So the antimicrobial, many researchers to validate the studies of this, as I already told you, without the validation of all the concepts, nowadays none of the things is being accepted. So on the Chitrak also many type of antimicrobial studies has been researches has been done to see the antimicrobial properties of Chitrak. The methanolic extract of the stem and the leaves of the Chitrak were tested on both extract, tested in both extracts showed the antimicrobial activity. Also, the stem extract was found to be more antimicrobial against the Pseudomonas aeruginosa and the Penicillium expansum species. The next study which shows the its antimicrobial effect is the leaf extract of Chitrak of Lumbago xylonica shows the maximum antimicrobial activity against both Staphylococcus aureus subspecies and Staphylococcus aureus and Fusarium oxysporum. So my motive to show these researches which I have told you now about all the toxic plants is that the concept, the effect of all these toxic plants on the different type of microbes is proved by many of the researches which has been done nowadays through different types of technologies. So if now those concepts have been validated, and now it's the time to switch over to the phytochemicals, so that the side effects which are being caused by the synthetic antimicrobial compounds can be removed, or we can treat the disease in a more efficient manner and become healthy, and no resistance bacteria can develop, and so that the treatment of the disease with the resistance of the bacteria, the treatment of disease becomes very, very difficult. So if we switch over to the phytochemicals, maybe we can treat the disease in a very well manner, and the development of the resistant bacteria can be reduced. So with all this, my presentation is over, and thanks to all. Thanks for patiently listening, and thanks again to the organizing committee. Thank you.
Thank you very much, Dr. Ritu, for giving a valuable information about the medicinal property and toxicity of plant Dhatura, Langli, Aak, Gunja, Bhallatak, Kupuli, Arand and Chitrak. एक्चुअली इन मेडिसिन को देने के लिए मुझे लगता है डॉक्टर्स को काफी सोचना पड़ता होगा कि मेडिसिनल ट्रीटमेंट जो है उनसे कहीं इसके साइड इफेक्ट आए या नहीं आए, और मोस्टली डॉक्टर्स इसको यूज भी नहीं करते। हम भी हमारी प्रैक्टिस में कई बार इस तरह के मेडिसिन को यूज करने से दूर भागते हैं कि इनके कई कॉम्प्लिकेशन नहीं आ जाए। डेफिनेटली आपके द्वारा दी हुई जानकारी जो है वो हमारे सबके डॉक्टर्स के लिए लाभदायक होगी। आपका कीमती समय देने के लिए धन्यवाद। भविष्य में भी आपकी सहायता इसी प्रकार मिलती रहेगी। अगेन वेरी वेरी थैंक यू। Now I am inviting Dr. Raghavendra Dubey. Dr. Raghavendra Dubey, Sage University, HY, Department of Pharmacy. Can you listen to me, sir?
Yes, sir. Good evening, sir.
Thank you, sir. Good evening. Very good evening, sir.
Good evening to everyone. And I think I'm very thankful toward you. You have invited me. I am thankful toward all audience, and I think it's a very intellectual session is going on. So can you give me the permission to share the screen?
Yes, sir, definitely, sir.
Yeah,
Yes,
I got, sir. So I think my screen has been shared, and so it was a very nice session by Dr. Thakur and Dr. Kapoor, and I think both the session has very versatile things. One is basically belongs to the allopathic, and the second one is quietly toward the herbal and toward the Ayurvedic session. And I think the question remains the same, which we want to have is how we will look toward the safe and effective antimicrobials. And because when we talk about the antimicrobials, we will find that more than 100 of antimicrobials are still available in the market. We are talking about the 6th, 7th, and 8th generation cephalosporin. We are talking about the herbal medicine or Ayurvedic medicine, which are already available. So now moving in that direction, I think my presentation is very crisp about the 15-minute session, and I will more concentrate toward the technical parts. So basically, briefly, I will review about the antibiotics. We know about the antibiotics. They are the vital life-saving medicine. I think the antimicrobial resistance is a global and local problem, and which is a very potential problem to address in the moment. We need to understand the link between the antimicrobial use or misuse with its resistance. And the most important thing is the antimicrobial drugs which we can use in this particular parts. So moving toward the antimicrobials, we already discussed. Thakur sir has given the brief about introduction about the antibiotics, about the antivirals, antifungals, and antiparasitic, all includes in antimicrobials. I'm just basically concentrating about the antibacterial. So antibacterial, when we talk about antibacterial, generally the mode of action already discussed in this previous session, maybe about inhibiting the cell wall or acting on the DNA. So basically any antibiotics break down the cell wall and stop the replication, or can inhibit or kill the microbes. And when we talk about the action of this antibiotic to the virus, we find there is no action of the antibiotic. So antibiotic should be very clearly, it only act on the bacteria, not on the virus. There is a different type of medication that we termed as antiviral, which are used for the virus therapy, or antiparasitic drug, which are used for the antiparasitic therapy. So the antimicrobials include all this. So when we talk about the antimicrobial, we need to understand that whenever we talk about the action of antibiotics on on the bacteria, we will find that once we will have a medicine prescribed, we usually take this medicine, and this medicine usually act on these microbes. They kill the microbes, and with the specific duration of time, we will find that most of the microbes will killed. And once the microbes will kill, definitely we'll get the suppression in the symptoms. We'll get the we'll find that the symptoms will be recovered. And this is the important period. The most of the people, when they will find that the symptoms are relieved, they stop the medicine. And this is a critical phase where we'll find that the resistance start developing. So if we want to completely eradicate this particular microorganism from our body, we need to take the full medical treatment of this antibiotics. Maybe it may be for 3 days, 5 days, or 10 days, as per the severity, as per the amount, as per the prescribed medicine, as per the dose, as per the type of medicine. So this full dose should require to be taken. Sometime it happens that some patients usually take only for 1 day or 2 days or for 3 days, not more than that. Once the symptoms get relieved, they stop the medicine, and this causes the resistance. The most important cause of this resistance in the antimicrobials, so is act due to this. So when we talk about the antimicrobials, we'll find there are two important categories which we find usually. That first is narrow spectrum, and the second is broad spectrum. So narrow spectrum basically act on a very specific and limited group of bacteria, and they are narrow spectrum also, and they are having low resistance also. But the broad spectrums are which act on the large group of the bacteria, but the overuse of this broad spectrum antibiotics usually cause the antimicrobial resistance. So when we talk about antimicrobial resistance, we need to think that the antimicrobial resistance occur when the parasites, virus, fungi change to protect themselves from the effect of antimicrobial drugs, which is designed to destroy them. So we usually plan the antibiotics to destroy this microorganism, but the microorganism get mutated with this particular dose. I already have discussed the previous slide that we are not taking the proper medicine, the proper duration of time, the proper dose of this medicine, and it will get mutated. So the World Health Organization has identified this as a big threat for the global health, because we are in a current age, we are talking about the 7th and 8th generation of cephalosporin. So this much generation, not the first generation antibiotic, the second generation antibiotic is also effective at that time. But the change in the mutation in the antibiotics has yielded, and we have resulted it to bring up to the 6th or 7th generation of antibiotics. So now when we talk about the antimicrobial resistance, we need to see how it occurs. So it occurs with the two important modes. The first mode is by the human itself, and the second mode is with the use of the carriers such as the animals. So whenever we usually give the antibiotics to the animals nowadays, it is very common to give the antibiotics to the animal. Not only the animals, we are giving to the aqua cultivation place also. So whenever we give the antibiotics to the animal usually develop the resistance of this bacteria in their guts. And whenever these animals are used directly by the humans in as their food material without handling properly or without cooking, that this particularly develops the antimicrobial resistance in the humans. Not only that, the excrete products of this animal, or maybe the water containing animal feces, when used with the food crops and without washing these food crops, when you are using these food crops, again this will become a source of antimicrobial resistance development in the human beings. So this is the one important part. And what we find that in human we fix the dose, but in animal we generally the lack of knowledge, lack of availability to the common people, they usually use the high quantity of antibiotics, and the resistance in the antibiotic develops with that space only. The second important with the humans, when human get entered in the hospital and get with this infection, and the infection get the secondary infections, and this infection when circulated with the general public or with this community, this is again a mode of being resistance microorganism to be spreaded in the community. So these are the basic two important modes which we need to look at for the resistance. When we talk about resistance, it is not about the single microorganism. We will talk about the Escherichia coli. So it is not about that once Escherichia coli get mutated, it only get mutated and it will spread. It will help other microorganism also, other bacteria also to get transferred this another gene in the horizontal gene transfer procedure. And once it occurs, it will spread to other microorganism also. So via this, you will find that the microorganism which get this particular genes, this mutated genes, it get spread with the other microorganism, it get multiply, and this gene multiplication cause spreading of this resistance microorganism. So this resistance organism do not destroyed with this particular antibiotics. So when we talk about the penicillin, as the sir has told, the first drug which is being found as a penicillin. If we talk about the penicillin, if we are giving to any microorganism, suppose it kills all the microorganism, it is susceptible, bacteria is highly susceptible to the microorganism, we will give that. But some little bit amount if remains with this develops the resistance in this microorganism. And once this resistance develops, this resistance multiplies to give the multiple copies of this resistance microorganism. The same things what I told, that once symptoms recovered, patient usually stop taking the antibiotics and completing the dose of the antibiotics. This is the biggest problem. So once he stops the antibiotic, again the microorganism which remains, it develops the resistance, and that resistance meanwhile cause the development of resistance antibiotics, microbes. So what we need to do is, we need to finalize this thing by choosing the right microbes, right antimicrobials, We need to choose the right patient to deliver the antimicrobials. We need to take the right time to deliver the antimicrobials. We need to fix the right dose, the right route, the right duration. And not only that, we need to have a documentation of this all with use of the pharmacovigilance. We need to document the effect of the medicine. We need to document the effect of the medicine on different type of patients, different category of patients, different respect to the time interval. We need to find out the different route of administration by which the drug is effective or may cause the resistance. These all documented should be distributed and addressed to the all, maybe to the professional pupils and maybe to the local people also. So while we're talking about the quality, we need to first ensure about the quality, equity, and safety. The quality means we need to keep up to date with the evidence of all the important events or important drug which are striving and implementing to that particular standards. So we need to come up with this particular thing first. We need to have a equity, because nowadays we can see that our India is very, very particular about this thing, because those who are having ability to bear the medicine, they are getting that type of medicine, or maybe due to the cultural linguistic background, there are different type of medicine or different type of medical treatment available to the patient. And this is again a problem, because antimicrobial resistance once developed in a patient, it can be circulated to the society irrespective of cost, irrespective of any linguistic background or a cultural background. So we need to take this responsibility to give the equity of this particular distribution of antibiotics to everyone. Then again, the last important which we discussed is the safety. The safety is to bring the acceptability and sustainable standard by giving it to the right patient at right antibiotic at right time at right dose with respect to right route of administration and right duration of action. So what type of approach we can use for this appropriate antibiotic use? We need to work with the health plan to monitor the prescribed habit, which is very, very important. We need to work in this way to have a correct planning for prescriptions about this medicine, not about the how with the sale of the drugs. We are basically working on the completing the self profit of the particular drugs. So, but not looking at that dimension, we need to see the health plan and the monitor prescription of the habits of the drugs. We need to collect the information of resistance pattern. This is again a very important, because if we see the if we will talk about currently the azithromycin is mostly used drugs now currently days. If you will find it before 5 years before the azithromycin is no more in existence for giving for the patient. So now we can need to understand what is the resistance pattern of the drug, and according to that we need to prescribe the medicine. So we need to collect the information about the resistance of that. We need to improve the diagnosis. It is very important. We need to have a very strong tool for identifying the resistance. Maybe it may be through the patch analysis, or maybe through culture and sensitivity assay. We need to identify the basically resistance pattern of the microorganism. And the most important thing, we need to come up with the education institute and with the professional organization, the medical professional, to educate each and everyone and to educate the public about the appropriate use. And nowadays WHO is daily telecasting. If you will see the radio also, WHO is daily informing about the use of antibiotics. So this we need to go with this awareness. The national plan already has given about this particular thing. We need to first important is we need to create the awareness, understanding and educating about this particular antibiotics and use of this antibiotic, and that should be very, very strictly according to the prescription. We need to work on the knowledge and evidence base about the surveillance of this antimicrobial resistance on humans also, on animal also, on the animal environment also. We need to create such type of laboratories for this. We need to work on the healthcare, that is by infection prevention and control. And not only that, we need to work on this optimum use, irrespective of any caste, religion or culture, or any on the basis of any other principles. We need to work for giving the right medicine at the right route. Then again, we need to work on innovation and R&D. We need to identify the health and safety of this particular medicine, the new innovation with the certain finance in this particular direction. We need to work on this. And the lastly, we need to have a collaboration, and not only the collaboration, we need to work jointly on the level of international, national, or maybe state level. We need to work for identifying such type of antimicrobial resistance, and we need to work to prevent the same. When we talk about the AYUSH medicine, the most important thing is not only about the treatment, it is about the primary prevention, and already given in the Ayurvedic text, we need to follow the same about the dincharya, the daily regimen followed by the individual, the ritu acharya, the seasonal regimen which is followed, the seasonal cleansing of the individual. Not only that, we need to work and guide about the non-retention of natural urges. We need to take the rasayanas to increase the immunity, and not only that, we need to have certain important thing to improve the mental strength using the yoga and dhyana. So this important thing should be bring in the public address, and this should be used to overcome this antimicrobial resistance. So there are a number of Ayurvedic formulation which are available, maybe Trifla Churn or Dashamula Churn, or maybe Maha Sudarshan Churn. These all churns are being already available. We need to come up with the awareness to the general public about the importance of this particular medicine in the treatment of the antimicrobial, in the case of complicated antimicrobial resistance case. Not only that, we need to work on immunomodulatory rasayanas. These rasayanas are strengthening the body innate defense mechanism to fight against this microorganism. So we need to come up to advertise these particular things, because we have already seen this in the corona period also. This in corona period, most of the immunomodulator has helped to overcome this particular disease, to get our immunity to be innate with this rasayanas and this Ayurvedic medicine. So we need to come up, the AYUSH need to come up for this helping in the dimension to have this antimicrobial resistance medicine. So number of medicine are already available. Already madam has told about the number of medicine. I will tell you about the, yes sir. Yes sir. So I'm finishing it with my conclusion point that there is a need of safe and effective antimicrobials, and we need to, for that we need to work on equity, quality and the safety of the antimicrobials. We need to optimize the use and plan of the action. This is very, very important, because we are having the medicine with us, but due to the lack of the knowledge of the use and action in the general public, there is a cause of antimicrobial resistance. Then we need to work more on preventive measures of the antimicrobial with respect to the Ayurveda. We need to work on these all 6 principles, and cumulatively we have to come up with certain research innovation for the leadership for the antimicrobial safety and working on the same. So this is all about from my side. Thank you very much for listening me.
Thank you very much, Dubey sir. Thank you very much for giving a valuable information about the AMR in human body and animals also, and the quality of the antibiotics, destruction of gene, correct information about diagnosis and treatment, sensitivity of the drugs. एंड डेफिनेटली आपने जो आयुर्वेद के बारे में बताया कि आयुर्वेद में जो पंचकर्म है, योगा है, दिनचर्या है, रात्रिचर्या, ऋतुचर्या, इन सभी का यदि पालन किया जाएगा तो निश्चित रूप से बॉडी की इम्यूनिटी बढ़ जाएगी, और यदि हम कोई मेडिसिन का यूज भी कर लेते हैं तो उसके दुष्प्रभाव हमारे सामने नहीं आएंगे। एक बात आपने बहुत इंपॉर्टेंट कही कि सारी पद्धतियों को मिलकर के काम करना चाहिए, क्योंकि हमारा जो पर्पज है वो मानव सेवा है। हम सभी मानव सेवा के प्रति प्रतिबद्ध हैं, इसलिए हम सभी मिलकर के इसमें आगे बढ़ेंगे और आगे काम करेंगे। आपका कीमती समय देने के लिए धन्यवाद। आगे भी आपका सहयोग इसी प्रकार प्राप्त होता रहेगा। धन्यवाद दुबे सर। नाउ आई एम इनवाइटिंग डॉक्टर मनोज अदलखा, असिस्टेंट प्रोफेसर फार्माकोलॉजी डिपार्टमेंट, डॉक्टर एसआर, राजस्थान आयुर्वेद यूनिवर्सिटी। आपको इनवाइट करता हूं। आप नेशनल इंस्टीट्यूट ऑफ आयुर्वेद से एमडी पीएचडी हैं। आपको पूर्व में भी बहुत सारी रिसर्च कार्यों के लिए, आपको शोध कार्यों के लिए अवार्ड किया जा चुका है। एमडी में अपने रिसर्च कार्य के लिए आपको स्वर्ण पदक भी मिल चुका है। इसके अलावा आप अपनी सेवाएं जम्मू कश्मीर में और हरिद्वार के दुर्गम इलाकों में भी दे चुके हैं। वहां पर विशेष रूप से सेवाएं देने के लिए इनको भेजा गया था। अभी जोधपुर और जोधपुर के आसपास के इलाकों में नशा मुक्ति एवं जितने भी ड्रग्स पाए जाते हैं उन ड्रग्स के मापदंड गुणवत्ता को देखने के लिए आप लगातार कार्य कर रहे हैं। वहाँ के आईआईटी के साथ मिलकर भी कार्य कर रहे हैं। वो डेफिनेटली आयुर्वेद में आपका एक तुलनीय योगदान है। नाउ आई एम इनवाइटिंग डॉक्टर मनोज अल्लक्का फॉर गिविंग द वैल्यूबल इंफॉर्मेशन अबाउट द मेडिसिन एंड एंटीबायोटिक्स, आयुर्वेद एंटीबायोटिक्स। वेलकम।
थैंक यू डॉक्टर अखिलेश। मैं ज़्यादा समय नहीं लूंगा फॉर्मेलिटीज में, और अपना जो टाइम है उसको ध्यान में रखते हुए बस आप इतना देखिएगा सर स्क्रीन।
यस यस यस
यस,
कंटिन्यू योर प्रोसीजर। वी कैन नॉट सी योर स्क्रीन बट W is moving. Yes, we can see.
Yeah.
Yes, doctor, continue.
Now, जैसे सर ने अभी बताया कि अपने जो आयुर्वेद की जो भी अपने रेजिमेन है, दिनचर्या है, ऋतुचर्या है, जिसमें जो भी अपने रसायन कर्म है, इनके द्वारा तो निश्चित रूप से अपन इम्यूनिटी सिस्टम को या हेल्दी रख सकते हैं। लेकिन इसके साथ साथ हमारे, जैसे मेरा डिपार्टमेंट ही मेडिसिनल प्लांट से रिलेटेड है, तो मैं आज अपने इस प्रेजेंटेशन में ये थोड़ा सा आपको बताऊंगा कि आजकल जो फाइटोकेमिस्ट्री और एंटी माइक्रोबियल टेक्निक्स है, इन विट्रो स्टडी है, जिसके थ्रू कि हम एंटी माइक्रोबियल स्टडी करते हैं डिफरेंट बैक्टीरिया पर या फंगल इन्फेक्शन पर या अदर, तो उसके थ्रू भी हम ये प्रूव करें हैं कि जितने भी हमारे हर्बल प्लांट से वो भी एंटी माइक्रोब्स पर किस तरह से एक्शन करते हैं। जैसे कि मैं आपको बताऊं, आयुर्वेद में भी आपने देखा है कि आचार्य ने अपने जो डिफरेंट आचार्य थे, चरक, सुश्रुत, ऑल आर मेंशन इन देयर टेक्स्ट दैट द वर्ड्स कृमि फॉर द फॉर द माइक्रोब्स। कृमि वर्ड उन्होंने ब्रॉड स्पेक्ट्रम में लिया है। कृमि से केवल वंश को नहीं लेना चाहिए, यहाँ पर उनका मीनिंग है आपके चाहे बैक्टीरिया हो, चाहे वायरस हो, या प्रोटोजोआ, या किसी भी तरह के माइक्रोब्स हैं, उन सब के लिए ये वर्ड यूज करा। और इसी को लेकर आयुर्वेद के तो सभी जानते हैं इस बारे में। मैं ये बताना चाहूंगा कि आयुर्वेद में भी जिस तरह से कि चरक ने अपने टेक्स्ट में महाकषायों का मतलब ग्रुप ऑफ 10 ड्रग्स के कॉम्बिनेशन का जो डिस्क्रिप्शन दिया है, उसमें कृमि महाकषाय मीन्स ये कृमि को लेकर यानी माइक्रोब्स के लेकर एक पूरा ग्रुप बनाया हुआ है जिसमें 10 हर्ब्स को बताया है। और उसको हमने ये भी देखा है कि हम अपने यहाँ पर एमडी स्कॉलर्स को रिसर्च करा के भी देखा है कि निश्चित रूप से उसके अच्छे रिजल्ट हमको देखने को भी मिले हैं। तो अपन देखते हैं कि जिस तरह से आप स्लाइड्स आर विजिबल। डॉक्टर अखिलेश, स्लाइड्स आर विजिबल? हेलो,
अभी रुक गया, अभी नहीं आ रहे।
No, not visible. Sir, not visible.
now okay,
Not visible.
Now it's okay, but
now it's okay.
we see only blank screen.
Okay, now
Not visible.
visible?
Yes, visual.
now
Continue with the topic.
yeah.
Now visible, sir.
Okay, sir. Okay, thank you. As you all know that most of antibiotic causes side effects due to increasing reactive oxygen species. इसके कारण से इसका जो मेन कारण है जो ह्यूमन बॉडी पे आरएस का पड़ता है, उस वजह से अपन ने देखा है कि ड्यू टू आरएस may cause damage to DNA, RNA and protein in the cell and also may cause cell death. So हम देखते हैं कि साइड इफेक्ट जो दिखाई देते हैं उसके कारण से और उसके कारण से ही इट्स प्रोड्यूसिंग कैंसर आल्सो। तो ऐसा नहीं है कि मॉडर्न के जो अपन एंटीबायोटिक जो भी यूज करते हैं कि सभी से इसी तरह के इफेक्ट देखने को मिलेंगे। उसका भी एक जो उन्होंने सिस्टम बनाया हुआ है कि अपन जैसे कि देखते हैं कि निश्चित रूप से जो एंटीबायोटिक मॉडर्न मेडिसिन में दिए जाते हैं, अगर वो लॉन्ग ड्यूरेशन तक अपन लेते हैं तो निश्चित रूप से ये सारी कंडीशन और साइड इफेक्ट देखने को मिलते हैं। इस कारण से जो हर्बल मेडिसिन है वो टोटली सेफ है और उसको यूज करेंगे तो मेरे को लगता है कि उससे साइड इफेक्ट कम देखने को मिलेंगे। नाउ जैसे मैं कुछ हर्बल मेडिसिन के बारे में बताना चाहूंगा जो
Slides are not moving, sir. It has been stuck up.
I think there is some technical problem, continued technical problems in Jodhpur. Sometimes we cannot see, sometimes we cannot listen the voice. Dr. Manoj. Dr. Manoj. Dr. Manoj, can you listen me? Hello, Dr. Saab. Site is not visible. Slide is not visible, as well as your voice. अगर स्लाइड में कोई दिक्कत आ रही है तो हम वर्बली कंटिन्यू कर सकते हैं, बिकॉज वी हैव लेस टाइम. The webinar should be completed at 9.
Is it okay if I can continue only by giving talk without slides?
How to?
Slide नहीं आ रही है तो verbally give any idea about the antibiotics in Ayurveda. We cannot see the slide.
Okay, I continue. था विडन के ऊपर, विडन के बारे में बता रहा था. इसको कृमिघ्न द्रव्यों में सबसे पहले चरक ने वर्णित करा है, और इसके जो एक्टिव प्रिंसिपल है उसमें एम्बेलिन, क्रिस्टेम्बेलिन और होमोएम्बेलिन एंड क्वेसर्टियोल, इस जो एक्टिव कंपोनेंट है ये चारों, इसके थ्रू ही ये अपनी एंटीमाइक्रोबियल प्रॉपर्टी को शो करता है, जो कि ई कोलाई, शिगेला, स्टेफाइलोकोकल, साल्मोनेला, पैराटाइफी बी पर भी इसका कार्य देखने को मिला है. और रिसर्चेस, मैंने वो स्लाइड शो नहीं हो रही है तो इसमें सारी रिसर्चेस शो की हुई है कि कहाँ पर ये रिसर्चेस दिखाई गई है. और इसके अलावा जैसे हम ये तो देख लेते हैं कि ये प्लांट कृमिघ्न है, लेकिन क्या इसके बारे में थोड़ी सी मैं जानकारी और देना चाहूंगा कि इसका जो एंटीमाइक्रोबियल एक्टिविटी जो इसका पार्ट शो करता है वो फ्रूट एंड रूट है. रूट एंड फ्रूट के थ्रू ये एंटीमाइक्रोबियल प्रॉपर्टी शो करता है, जिसके की थेरेपेटिक डोज 3 to 5 ग्राम ऑफ पाउडर है. और मार्केट में जो इसके प्रोडक्ट अवेलेबल हैं उसमें संजीवनी वटी, पंचनिम्बादी चूर्ण, अमृता गुगुल आर दे सम ऑफ इट्स वेल नोन फॉर्मुलेशन इन द मार्केट. नाउ क्लोव, जैसे कि आप सब जानते हैं, मैं कुछ ऐसी मेडिसिन, कॉमन मेडिसिन के बारे में ही बता रहा हूँ जो आप सभी जिससे परिचित हैं, जैसे कि लवंग, साइजिगियम एरोमेटिकम, ये भी एंटीमाइक्रोबियल एंड एंटीऑक्सीडेंट हेल्प से, और इसके जो एक्टिव प्रिंसिपल है उसमें जो यूजीनोल है उसके थ्रू ही ये एंटीमाइक्रोबियल एक्टिविटी शो करता है. और रिसर्चेस इसमें मैंने शो की हुई है, जो कि दिख नहीं रहा है, मैं इसको बाद में शेयर कर दूंगा आप सभी से. इसी के कारण से, यूजीनोल के कारण ही ये डिफरेंट बैक्टीरिया ग्राम पॉजिटिव और नेगेटिव पे अपना इफेक्ट दिखाता है, जिसमें मुख्य रूप से लिस्टेरिया मोनोसाइटोजीन्स, क्लेब्सिella न्यूमोनियाई, एस ऑरियस, ई कोलाई आदि बैक्टीरिया हैं, जिसके कारण से ये अपनी एंटीमाइक्रोबियल एक्टिविटी शो करता है. एंड आल्सो क्लोव हैज स्ट्रॉन्ग एंटीऑक्सीडेंट इफेक्ट दैट कैन न्यूट्रलाइज आरओएस एंड अदर फ्री रेडिकल्स इन लिपिड चेन, देयरफॉर दे इनहिबिट फर्दर ऑक्सीडेशन ऑफ द लिपिड्स. इसके थ्रू ही ये अपनी एंटीमाइक्रोबियल और एंटीऑक्सीडेंट प्रॉपर्टी दिखाता है. ये इसका मैकेनिज्म ऑफ एक्शन है. नाउ नेक्स्ट, टर्मेरिक, जैसे कि आप सभी जानते हैं, हरिद्रा, कर्कुमा लोंगा, well known Ayurvedic herbs which shows its role on microbes. और इसमें भी मेनली जो इसका एक्टिव प्रिंसिपल है कर्क्यूमिन, वो ही एंटी माइक्रोबियल प्रॉपर्टी रखता है. बाकी भी रखते हैं, लेकिन सबसे ज्यादा जो प्रॉपर्टी हमको रिसर्चेस में देखने को मिली है कि कर्क्यूमिन के थ्रू ही ई कोलाई, एस ऑरियस, साल्मोनेला टाइफिम्यूरियम, स्यूडोमोनास ओरिजिनोसा, इन बैक्टीरियाज पर इसकी रिसर्चेस देखने को मिलती हैं कि ये एंटीमाइक्रोबियल एक्टिविटी शो करता है, साथ में एंटीऑक्सीडेंट तो है ही। इसके अलावा रिसर्चेस ये भी शो करती हैं कि एंटीवायरल the researchers shows antiviral activity of this plant against HIV virus also, means ये बैक्टीरिया और वायरस दोनों पर ही अपनी एक्टिविटी शो कर रहा है। ये सभी रिसर्चेस मैंने शो की हुई है इसमें। देन दारू हरिद्रा जो कि हाई एल्टीट्यूड का प्लांट है, हो सकता है सभी नहीं जानते हो, हिमालयन रीजन में ये प्लांट पाया जाता है, बर्बेरी सेरिस्टाटा। इसकी 13 से ऊपर स्पीशीज पाई जाती हैं और लगभग सभी में ही बर्बेरिन एल्केलॉइड पाया जाता है जो एंटीवायरल है और एंटी बैक्टीरियल है। और बर्बेरिन एल्केलॉइड के कारण ही ये वायरस पे तो इफेक्ट दिखाता ही है, साथ में स्यूडोमोनास ओरिजिनोसा, स्टेफाइलोकोकस ऑरियस, क्लेब्सिएला न्यूमोनाई एंड ई कोलाई कैंडिडा एल्बिकेंस, इन सभी पर रिसर्चेस हुई हैं इसके बर्बेरिन एल्केलॉइड को लेकर। जो साथ में अपन ने देखा है जैसे कोविड में भी जो इस ड्रग ने बहुत अच्छा काम कर रहा है, और साथ में हम देखते हैं कि लीवर डिसऑर्डर में भी इसका बहुत अच्छा रोल देखने को मिलता है। वैसे अपन मार्केट में इसके जो प्रोडक्ट देखेंगे तो वो दारव्याधि क्वाथ है, दारव्याधि ले या दारव्याधि तेल, इन सभी की एंटी माइक्रोबियल एक्टिविटी शो होती है, तो आप इसको यूज भी ले सकते हैं अपने ट्रीटमेंट में। नेक्स्ट वेल नोन ड्रग आमलकी, जैसे कि आप सभी जानते हैं कि आमलकी पे भी बहुत ज्यादा रिसर्च वर्क हुआ है, और इसके भी जो एक्टिव प्रिंसिपल है उसके थ्रू ही इसने जो अपने रिसर्चेस में जो अपने एक्शन शो करे वो बैसिलस पे कराएं, स्टेफाइलोकोकस पे कराएं, क्लेब्सिएला, ई कोलाई, विब्रियो कोलेरा, कैंडिडा एल्बिकेंस, शिगेला, एंटेरोकोकस और आदि बाकी और भी एंटी फंगल एक्टिविटी पर भी इसका देखने को रोल मिला है इस वाली ड्रग का, अमलकी का। और इसका जो यूजफुल पार्ट है जो एंटी माइक्रोबियल एक्टिविटी शो करता है वो फ्रूट है, जिसके की फॉर्मुलेशन मार्केट में अवेलेबल है, जैसे च्यवनप्राश है, त्रिफला चूर्ण है, भ्रम रसायन, धात्री लो, धात्री रसायन आदि सभी में ही आमल की देखने को मिलता है। इसका पाउडर है इसके अंदर, और बाकी भी है प्रोडक्ट इसमें, लेकिन मार्केट में जैसे अभी देखते हैं अपन च्यवनप्राश का भी कोविड में बहुत अच्छा रोल देखने को मिला है, वो इसी कारण से मिला है कि एक्चुअली रेजुवेनेटिव प्रॉपर्टी तो ये रखता ही है, साथ में ये एंटी माइक्रोबियल प्रॉपर्टी भी रखता है।
Doctor sir, please conclude within 5 minutes because next webinar will be start at 9. We are sorry.
Okay. Now एक और ड्रग के बारे में बता देना चाहूंगा जो आजकल बहुत ज्यादा इंपॉर्टेंट है, वो चल भी रहा है, कैंसर में इसका बहुत अच्छा रोल है, इसकी मोरिंगा ओलिफेरा का। मोरिंगा ओलिफेरा के ऊपर भी बहुत ज्यादा रिसर्चेस हुई हैं, और ये भी अपनी एंटी इंफ्लेमेटरी, एंटी बैक्टीरियल, हिपेटोप्रोटेक्टिव एंड एंटी कैंसरस एक्टिविटी शो करता है, और साथ में जो अपनी एक्टिविटी शो करता है वो स्टेफाइलोकोकस ऑरियस, स्यूडोमोनास ओरिजिनोसा, ई कोलाई आदि बैक्टीरिया पर भी ये अपनी एक्टिविटी शो करता है। मार्केट में भी इसके श्यामादि चूर्ण, शोभाजन आदि लेप आदि का यूज किया जाता है। इसी तरह से हम देखते हैं कि नीम है, सिनेमोन है, जिंजर है, जिंजर के बाद अपन जैसे मैं अब थोड़ा सा टाइम कम है, नहीं तो मैं आपको इन सभी के बारे में डिटेल में बताता कि अपने किस किस एक्टिव कंपोनेंट के थ्रू ये एंटी माइक्रोबियल एक्टिविटी शो करते हैं। अंत में मैं ये कहना चाहूंगा, जैसे कि टाइम की कमी है, वैसे ड्रग्स की लिस्ट तो बहुत लंबी है, मैंने डॉक्टर अखिलेश को और सर को अपना पीपीटी शेयर भी करा है, तो आप अगर लेना भी चाहें तो उनसे ले सकते हैं। इसमें ये सब कुछ बताया हुआ है कि जितने भी हर्बल प्लांट्स हैं ये केवल ऐसा नहीं है कि ओनली फॉर डाइटरी पर्पस अपन इसको यूज कर रहे हैं, अपन इसको मेडिसिनल पर्पस से भी यूज करें, और निश्चित रूप से इनके ऊपर जो अपन पहले जो कमी थी कि इनके ऊपर रिसर्चेस बहुत ज्यादा देखने को नहीं मिलती थी, लेकिन प्रेजेंट टाइम में हम हमारे स्कॉलर से भी और CCRS भी इसमें लगा हुआ है, और एंटी माइक्रोबियल रिसर्चेस अब हमारे जो मेडिसिनल प्लांट्स हैं इस पर बहुत ज्यादा हो रही है, और आप चाइल्डलाइन को चेक भी कर सकते हैं, और डिफरेंट बैक्टीरिया पर इनके इफेक्ट मिलते हैं। तो मैं ये कहना चाहूंगा कि ये सेफ है और इनको मैक्सिमम यूज करना चाहिए। थैंक यू।
Thank you, Dr. Manoj, for giving information about the Ayurvedic antibiotics, just vidang, lavang, and the effect of microbe, turmeric पर आपने बताया कि HIV पर इसका काफी अच्छा काम है, चिगरू के बारे में बताया कि आपने कैंसर के रोगियों में अच्छा लाभदायक है, दारू हरिद्रा के बारे में जैसा आपने बताया, और डेफिनेटली मुझे सुनकर और आश्चर्य होता है कि आप जैसे विद्वानों के द्वारा कि हम जो हमारी आयुर्वेदिक मेडिसिन है वो कितनी इफेक्टिव है, और उसमें उनमें जो जीवाणु मारक क्षमता है वो कितनी सारी है। हम व्यर्थ में ही बहुत सारी केमिकल एंटीबायोटिक्स के बारे में लोगों को बताते हैं। हमें आम लोगों को बताना चाहिए और खुद भी ये समझना चाहिए कि हमारी जितनी भी दवाइयां हैं, जितनी भी आयुर्वेद मेडिसिन है, वो कितनी इफेक्टिव है। डेफिनेटली भविष्य में जनता इनका यूज भी करेगी और हमें इनसे इसके प्रति जागरूकता भी लोगों के बीच में ले जाना चाहिए। थैंक यू वेरी मच डॉक्टर मनोज, आपका कीमती समय मिला। आगे भी इसी प्रकार आपका सहयोग प्राप्त होता रहेगा, ऐसी आशा है। निश्चित रूप से आज का जो वेबिनार है बहुत इंपॉर्टेंट था और उसका टॉपिक भी बहुत इंपॉर्टेंट था, क्योंकि आज के समय में हम देख रहे हैं कि भारत सरकार और प्रांत की जो सरकारें हैं वो इसके लिए गंभीर है। बल्कि दिल्ली ने तो एक आदेश भी निकाला था कि जितने भी डॉक्टर जो प्रेस्क्रिप्शन लिखते हैं उन प्रेस्क्रिप्शन पर नजर रखी जाएगी कि वो जो एंटीबायोटिक लिख रहे हैं वो बीमारी के लिए है भी या नहीं, उसका समय कितना है, या क्या व्यर्थ में तो नहीं लिखी जा रही। क्योंकि अभी डॉक्टर राघवेंद्र दुबे ने बताया कि यदि हम एंटीबायोटिक को ज्यादा मात्रा में लेते हैं तो हमारे जीन के लेवल पर भी चेंजेस कर सकती है। कितनी बड़ी बात है, जीन के लेवल का चेंज होने का मतलब है कि जो बीमारियां एक पीढ़ी से दूसरी पीढ़ी में भी जा सकती है। बहुत ही गंभीर विषय है और इस पीढ़ी को WHO ने तो कहा है कि जो मुश्किलें हमारे सामने आ रही है उन सबसे बड़ी मुश्किलों में आज एंटीबायोटिक का मिसयूज है। हम सब आयुर्वेद के डॉक्टर्स मिलकर के WHO की जो समस्या है उससे सोल्व करने में अपना बहुत बड़ा योगदान कर सकते हैं। थैंक यू वेरी मच डॉक्टर मनोज। सर, डॉक्टर पवन सर,
thank you very much, Dr. Akhilesh.
completed. We have completed our
Thank you very much.
now
During this COVID pandemic, we have observed that antimicrobials have been failed serial wise.
Definitely
3 months, some antibiotic, again 3 months, some again next antibiotic. So that experimentation, trial and error method, if could have been done with Ayurveda, definitely the loss of lives were too less, too smaller in number. That is my confidence on Ayurveda.
Yes.
Thank you the speakers for 499th webinar on Ayurveda AYUSH, the best and safe effective antimicrobials. Thank you, Dr. Akhilesh, for coordinating, organizing and moderating. Thank you very much. So nice of you. I would like to welcome Dr. Premdani ji from welcome sir. Welcome.
Thank you, sir. Thank you.
Welcome from Netherlands.
Yes.
Welcome, sir.
Welcome, sir. Welcome, India.
Thank you.
Dr. Namrata Jangam from Netherlands. Welcome, madam. Dr. Peperina, Holland. Dr. Harish Verma ji from Canada, Toronto. Welcome, sir. Just we are going to start our Global AYUSH series, 500th webinar. This brainstorming journey, AYUSH SAMRIDDHI, is now achieving by this webinar 500th milestone, sir. Welcome, sir. I hope Dr. Harishankar Sharma ji will also join very soon. How are you, sir?
Harishankar Sharma, sir, from Japan.
Netherlands. Dr. Harishankar Sharma, sir, from Netherlands. Welcome, Girish, sir. Welcome, Dr. Girish Momaya ji. Welcome. Very senior expert from Meru, Maharshi Ayurved European University. Welcome, sir. Welcome.
Very pleased to be here.
Glad to listen to you, sir. Glad to listen to your blessings today.
Swamiji, we cannot see you.
Yeah, I'm trying. Now it should come.
Yeah, now I can see you, Swamiji.
Yeah.
Swagatam, Swamiji, Swagatam.
Swamiji, namaskar. Professor Dr. Vishnu Sharma from Assam, Guwahati.
Very pleased to see you and meet with you.
Yes.
Namaskar. Myself, Dr. Akhilesh Bhargav from Indore, Madhya Pradesh, the cleanest city of India, 6 times.
Beautiful. Yes.
Myself, Vaidya Rajan Patenkar from Mumbai.
Namaskar.
Guruji, pranam.
I am Dr.
Vineet
Rajesh
Agnihotri
from
from
Sri
Haridwar,
University,
Uttarakhand.
Indore.